On behalf of Cambridge Health Tech Institute's Global Web Symposia series and our sponsor, Sentry Sciences, LLC, I'd like to welcome you to Particle Sentry AI, combining AI and flow imaging microscopy to analyze and monitor biologics. My name is Elizabeth Lamb, and I'll be the host and moderator for today's event. Now I would like to introduce our presenters for today. First is Chris Calderon, PhD, President, URSA Analytics. And our second presenter is Sandy Hutchison, Director of Software Products with Sentry Sciences, LLC. Welcome, Chris. The presenter ball is yours.
Thank you, Elizabeth, for the introduction. And thank you, everybody, for taking the time to listen to our presentation. Before I start, I'd like to acknowledge my experimental collaborators, the Ted Randolph Lab at University of Colorado Boulder, as well as a Project Charo and colleagues at the FDA are participating in, as well as Dean Ripple at NIST, and of course the team at Sentry Sciences. We've been jointly developing the software product that implements the methods that we'll be showing today. So, in this talk, we are ultimately interested in showing new methods to quantitatively characterize protein aggregation in biologic products. I'm going to start by describing some background to this problem. The two main areas I want to start focusing on are protein aggregation and flow imaging microscopy. I'll then briefly introduce convolutional neural networks, or CNNs, and show what problems these methods have tackled in modern computer vision. After that, I will demonstrate how variants of these algorithms can be used in helping characterizing protein aggregation measurements in some real-world applications. Throughout, I'm going to demonstrate high-level the machine learning algorithms like CNNs can be used in various pharmaceutical applications. For those interested in more of the technical details, feel free to email us, but there'll also be references provided in the talk throughout. So why do we care about protein aggregates? Protein aggregates in injected biologic products can look like foreign invaders to the body. When a biological product contains protein aggregates in the injected patient, this can result in a reduced efficacy of the drug over time, or worse yet, it may even result in a severe adverse drug response. Unfortunately, there are many ways in which protein aggregation can form in a product. Various stresses encountered in manufacturing, shipping, or administration, or just care in the hospital, how the drug has been stored, can lead to particle formation. In general, exposure of protein solutions to interfaces often causes particles to form, but freeze-thawing, pH changes like in a viral clearance step in manufacturing, mechanical shops, and shipping, all of these can result in particles. But the important thing to stress is the particle type of a protein aggregate depends on the applied stress. And here's just a cartoon showing two ways in which a particle can form from a protein monomer. In the top row, you have a cartoon of a simple monomer that basically grows by monomer addition. But you can also have agglomeration where small little particles form that have a little more secondary structure, and they aggregate and form these other fluffier particles in the agglomeration process. And there's a whole bunch of other processes in which particles can form in protein aggregation. The take home is that no two protein aggregates are the same. I like to say that they're snowflakes. There's a wide diversity of these protein aggregates when they're formed by different stresses. But the fortunate thing is that they have measurable properties. They all have different densities, shapes, size, refractive indices. And these are all the types of characteristics that you get from these protein aggregates depend on the stress that produce them. And fortunately, since a lot of these properties that we listed here before are reflected in images, you can use light microscopy to characterize these protein aggregates. And that's the theme of today. One tool we found to be particularly useful in characterizing protein aggregates is flow imaging microscopy. It's a backlit bright field technique which uses a microfluidics flow cell and a small volume of liquid from a sample of a protein product. And from a small sample, about 250 microliters, you can get from a stressed protein particle 10 to the third or 10 to the sixth images of particles from that formed in that particular container. The technique is very commonly used in characterizing biologics and formulation, but it's also being used as a compendial method it's being used as a orthogonal method to compendial methods, and it's also being explored for lot release. So what we're trying to do is try to push the utilization of the information content from these types of measurements. But I do want to stress that flow imaging microscopy is not the only technique our approach is applicable to. We can use any imaging technique that can characterize particles from protein solutions. For example, background membrane imaging or imaging flow cytometry with fluorescence. And these are in principle also applicable to the method that we're talking about today, but most of our work to date has focused on flow imaging microscopy, so that's where I'm going to focus today's efforts. So shown here is an image of a part of a flow imaging microscopy collage that was measured from a vial that was stressed, a MAB vial that was freeze-thawed. And then what we did was we pushed that thing through the flow imaging microscopy device, and we measured a large collection of images. And you can look in here, there's a very heterogeneous collection of images in this particular set. The question is, how do you characterize these particles? Historically, people have used count and size primarily as the way of summarizing the information content in here. But think of this, a picture is worth 1,000 words. There's a lot of information about the stress history encoded in this zoo of particles that we're looking at in this flow imaging microscopy data set. And subject matter experts know that there are better ways of using the information that's contained in these images. We're throwing away a lot of information when we just look at count and size. And the way that this project got started was thinking of methods for trying to utilize the rich information content in these high throughput flow microscopy devices. So as I said, subject matter experts know that there's a better way of characterizing proteins, but the differences in these images are very subtle. So shown here is a collection of MABs on the top row that experienced two different stress histories. One was freeze-thawed, and on the right, one was mechanically agitated with a viral clearance step. And if you look at these, just at quick glance, they don't look all that different. And on the bottom row, we have a polyclonal antibody, or IVIG, that was put through two different pumps. And the pumps were supposed to represent peristaltic pumps that would be representative of two different operating plants. We get two different types of images from the two different pumps, even though the underlying product's the exact same. So it's kind of the manufacturing history that this particular product experienced. Graduate students actually suffer through looking at these images, and they can actually intuit and figure out what some of the differences are. They can start to look at a large enough quantity of these images and make a best guess as to where they came from, but they have a very hard time quantitatively describing what features they're using to make their decision. But eventually, if they see enough images, they can kind of just make a best intuitive guess without a very quantitative algorithmic description. And that's what motivated us to actually come up with a automated algorithmic description for being able to distinguish these stress histories and summarize the information content in these flow imaging microscopy images. Aggregates aren't supposed to exist in a product, but they always do exist. But the theme of this feature is that, of this talk, is that bug can become a feature. And what we're going to be doing is showing how high-throughput optical microscopy combined with deep learning or artificial intelligence applied to protein aggregate data can assist in both formulation, looking at container closure screening, for example, quality control, and process analytical technologies. So the workhorse tool that we're going to be using throughout is something called a convolutional neural network. You may have heard about this tool from pop culture, Wired magazines, Popular Mechanics. For those of you who haven't, it's really a nonlinear transformation tool, and convolutional neural networks are very good at processing images. And so what is an image to a computer? It's really just a collection of pixels and a certain number of channels. If you have a color image, it's 3 channels, red, green, and blue channels, and they're all just pixel intensities between 0 and 255. So it's a collection of numbers that really just summarize what your cat looks like. What we're doing is, this is basically an array that we're going to be processing through a technique called convolutional networks, and I'll walk through exactly how this works at a high level in the next couple of slides. But then it basically extracts features for trying to figure out what is a cat-like feature, or what are the features that are encoded in this image, then passes that down in a feed-forward fashion to a classifier, like a logistical regression classifier. And the most popular application of this tool is this kind of classification tool, which takes an image and tells you, is there a dog or a cat, or some other object of interest in this particular image. The technology's been around for a very long time. It was developed before the 90s, but it became popular in the early 90s when it was used in optical character recognition. Think about a tool for automatically reading information from checks. But it really took off around 2015 when GPUs were able to be fine-tuned to make this process much more computationally efficient. And it's just exploded ever since. And so what did it do? Before in computer vision, this was a long-standing problem. People thought that it would be hard for a computer to distinguish reliably between a dog and a cat. And why is that problem hard? I can show my 3-year-old pictures of dogs, a couple pictures of dogs and cats, and they will be able to extrapolate what another new picture of a dog or a cat looks like. For a computer, that was hard in the past when you didn't use convolutional neural networks. A lot of people would use hand-engineered features. And so they would say, well, dogness is really a quality. Dogs have smushed noses. Well, grumpy cat over here has a smushed nose. Cats have pointy ears. The husky has pointy ears. The point is it's hard for a human to articulate a very robust feature that can be used in a predictive fashion. But convolutional neural networks are a data-driven technique where you put in a large number of dog pictures and a large number of cat pictures that are labeled, and you extract features that try to quantify dogness or catness in the images, and they can actually get about 99% accuracy in that type of problem. And so how does it work at a very high level? What you do is you have your input image, which is, again, just remember it's a collection of pixels with 3 channels. You estimate these small filters or kernels. So you say that here's a dog picture. You've got millions of dog pictures. You have millions of cat pictures. You try to estimate the parameters of these filters or kernels for what these are used are is kind of think of it as a dot product. These are all parameters. Each of these parameters are pixel intensities, but what you do with this one filter is you take this filter, you estimate the parameters that best try to characterize dog and catness in this example. You get these values, you then convolve that, or it's basically a dot product for images. You take this pick value, dot it against the pixel values, slide the window and convolve it. That's the operation of, that's the C part of the convolutional neural network. And you make this thing called an activation image. But you don't just do that for one filter or one kernel. You estimate a really high-dimensional model. You estimate a lot of filters and a lot of kernels. So here's the result of using another filter applied to the same image, and you get a very different activation image. And so that's the name of the game here. You estimate a huge number of kernels in the first layer, apply it to the input. You get one image for each kernel, an activation image, and this represents about 12 activation images shown here. So what you're really doing is you're taking an RGB image and you're transforming it to a higher dimensional image. These still have some dog-like features. You can kind of see that the human can make out somewhat what some of these objects are. But you're increasing the number of channels and you're doing this kind of in a feed-forward fashion. So you've taken one image that has 3 channels and now you have 12 channels. So that's the way I like to describe this technology. But then you basically rinse and repeat this procedure over and over again. So you took your dog here, you estimated your filter parameters, you get a new image. This is just treated as in the eyes of a computer, this is just treated as another image with more channels. You repeat the exact same process. You estimate a collection of filters, and for each filter, you get a new activation image from this input. And then you keep doing that procedure over and over again. And as you do that, the number of activation images tends to increase as you go through these feed-forward neural networks. And the human interpretability starts dropping off relatively quickly. The other thing to note in these images, you see these kind of blank spaces where there's just all blue. This means that in the layer above, there was no match for the particular kernel that the thing was applied to. So it's basically a 0 dot product. And as you go further and further in the neural network, more of these kind of 0 pattern activation images show up as a kind of a sparsity pattern. That's a common trait in simple neural networks. But the end goal is really to transform an image into a high-dimensional hyperspectral image. So each of these patches are new activation images. And this is what the computer uses to try to predict what a dog or a cat is. It doesn't use the original input images. It does this series of transformations to form this representation of the dog. Here's what a human would see. Here are 4 dogs in the eyes of a human. Here's what you transform them to in a convolutional neural network. Here's 4 cats. The task here is to separate dogs and cats. A human cannot interpret this information very easily, but if you start to compare the representations between the dog and the cats that we just showed, you start to see systematic patterns in terms of both the sparsity and the activation images show up. And that's what the computer does. It basically takes a high-dimensional feature vector and predicts class probabilities based on these inputs. And this is the real success story of modern convolutional neural networks. You have a large collection of labeled images, and it's able to very accurately distinguish between subtle things that a human can easily do, but a computer has had a hard time doing in the past. And it can do it with incredibly high accuracy. So back in 2018, or essentially 2017, we were interested in seeing if this technology could be applied to flow energy microscopy data. The difference here is that the images are not easily human interpretable. A subject matter expert sometimes even has a hard time looking at these images and determining exactly what's what. But we wanted to see how accurately we could use this same idea on labeled stresses from a monoclonal antibody stressed in 2 different ways and the IVIG that we showed before from 2 different plants. And the results we got were pretty shocking initially. What's shown here is we trained a neural network using 50,000 images of each of those types we showed before, the 4 classes that we had where we had the IVIG pumped with 2 different stress histories, the viral clearance and shaking step, the freeze thaw with the monoclonal antibody. And we trained a network to do this kind of classification to predict which class it is. We then took 10,000 test images that the neural network had never seen. And what we did is we know that the true label here is our pump A IVIG particles. And what this says is 70% of the time in that 10,000 in that collection of test data that we looked at, we correctly labeled the images that came from pump A as pump A. And the same thing for these other classes where we had IVIG and the monoclonal antibodies. And I want to stress this is with a single flow imaging microscopy image. And a perfect classifier would have a 1 across the diagonal of this so-called confusion matrix. But it's still doing a very good job with a single image of these really heterogeneous, hard to understand images of protein aggregates. You're getting 70% accuracy. And the other thing to note here is you don't get confused between polyclonal and monoclonal antibodies as well. So this with a single image was pretty amazing. But in pharma, you don't ever make a decision based on a single protein aggregate image. You often have many of these, even in a fill finish operation, you still have hundreds of particles that you might want to look at. So the idea that we had in this early 2018 paper was, well, you can actually just do a pooled estimator, making the assumption that all the particles come from the same stress history. And when you do that, if you only have 50 images of the particle, you get a nearly perfect classifier, which was great to us. We were really excited about that because the human accuracy by a subject matter expert was believed to be about 60% at best. And so we're doing a really good job with classifying. However, fundamental problems exist with this. This is a supervised learning approach where you have a huge number of collection of images that are labeled, and then you're just basically trying to predict a class label, for example, IVIG from pump B. But this problem is, it's also applicable to the dog and cat problem. So I'm going to use a more easy to understand set of images to show you how what the issues are with this kind of supervised classification approach and why it's a problem for a pharmaceutical application. So imagine what our neural network is just basically a black box technique. And what the thing really does is it takes an input image, processes it, and then predicts the class probability of this thing being a cat or a dog. And the 2 classes we're looking at here, there's only 2, so these probabilities have to add up to 1. And so it's 95% confident this thing's a cat. The thing's working wonderfully. Dog, same thing. We get a new image of a dog. The neural network is fairly confident this is a dog. We pass in a slightly harder dog that has some cat-like features. Huskies have eyes that could be considered cat-like and pointy. The neural network doesn't really care about human interpretable features, but its confidence is still very high, but it's gone down slightly. But this is kind of behavior that we expect. But if we train this neural network only using dogs and cats, but then throw in a few monkey wrenches, imagine we throw in a hyena. A hyena is not technically a dog or a cat. It's got a lot of features of both. And this is a failure mode that we'd want to see in a classifier. We put in an image that was unknown from a completely different class that the network is not supposed to do a good job training on. But what it says is I'm very confused. It's basically a coin flip between a dog and a cat in this particular case. However, suppose we put in a picture of a snake. This picture of a snake is a huge outlier, but the neural network needs to assign class probabilities. It's never seen a snake. It needs to say, is it dog-like or cat-like? The probability of the cat's 92%. This thing's overconfident that this is a cat, even though we know it looks nothing like a cat. And so this type of failure mode is well known in the literature, and it's a problem with these kind of supervised classification techniques where you have to predefine your labels. And it's also a problem in manufacturing. You don't know what's going to go wrong in your process. Lots of things can go wrong. So preparing for every possible unknown is very unrealistic. But we want to try to solve this problem. When you have new novel images that show up, we would like to be able to identify them. And that's what motivates the technique that we're going to be showing today. It's this fingerprinting technique that we published in 2020 in the paper shown below. But I'm going to walk through the basic ideas of it at a high level and then show a variety of use cases of the technology. And so before, what we were showing were convolutional neural networks where you were basically extracting features and then putting it into a classifier. The same technology is being used here in order to do image compression. So you could use the exact same kind of ideas from the convolutional neural network, but your objective function just changes. So instead of trying to find a class probability, what you're trying to do is to take a large collection of pixels that are spatially correlated and have different channels and compress that down to a 2- or 3-dimensional number that is informative. And so that's what we're doing here. So to illustrate the idea that's slightly different than classification is suppose you had something that's a little harder than dogs and cats. You had grayscale images of fashion accessories. I'm an engineer and I have a hard time distinguishing between some of these technical fashion accessories. The difference between a top and a pullover is a little fuzzy to myself. But this neural network, if you give labels to this thing, and instead of trying to get a class probability, you try to compress these grayscale images, which are 32 by 32 in this particular case, so that's about 900 pixels, and you try to compress it down to a 2-dimensional number, you start to see all the different fashion accessories form these clusters or these embeddings where they're all the fashion accessories from one particular class all kind of clumped together. Even though it's hard to say exactly what makes a top and a pullover different, this technique said sometimes they bleed over, but you can do a pretty good job in using 2 numbers to say this is what the essence of a pullover is in this particular case and all the different fashion accessories. And so it's an image compression scheme. The same idea can be applied to the data sets that we showed before with the monoclonal and polyclonal antibodies with different stress histories. So shown here are the IVIG, and shown here are the monoclonal antibodies, 2 different stresses. They form these 2 different islands. Within these islands, they have some overlap between the monoclonal and the polyclonal, but they're pretty well separated with 2-dimensional number. So again, what I want to stress what we're doing is how we form, what a fingerprint is and how we form it. So now that we've trained a neural network that can compress images, great. What do we want to do with that? Well, what's the point? So here's one image from a trained neural, we've trained the neural network and we get a new image of, say, freeze-thaw monoclonal antibodies. And here's one image, we compress it down to a point. We get another image, compress it down to a point. The idea is that we have a reference condition or a baseline. So you can imagine having a particle, a set of particles from a fill-finish history of when you're sampling your production line. Or you could just have, you're doing formulation and you're interested in looking at container closure interactions. And so you have a large number of aggregates from a particular container closure that were stressed in a certain way. But you just have one type of reference condition that you're interested in looking at. Once you have a large reference condition and you collect a large number of images from that reference condition, what you can do is then apply that compression scheme to every single image in there, and you basically get this scatter plot, or this large collection of points that eventually kind of spill out a systematic pattern. And what the fingerprint is, it's really just a fancy word for a delicate word for a probability density function. But what we're just really trying to do is estimate the probability density function of this collection of scatter plot points. So what we're imagining is if we had an infinite collection of these images, what would be the histogram or the probability density function that would result if we had an infinite collection of particles? And that's the function we try to estimate. So this function, which is really just a probability density function, again, remember, think of something that integrates to 1 if you have a 2-dimensional surface. This is a contour, so the most highly probable region, the mode of this PDF is shown right here. But the PDF is really what we call our fingerprint. So it's a statistical summary of the collection of scatterplot points that result from compressing all these images down to a 2-dimensional number, and then this is the distribution of those points. It's a quantitative description of what we have. I realize that this slide is really busy. Sandy is going to speak to it later in the product component of this talk. But what I want to do is I want to pick out a couple of use cases in pharma to show how this technology can be, how we envision it to be useful. For example, I'm going to be showing a container closure study, as well as a process monitoring study and a shipping study in our particular case. So I'm trying to sample things from upstream to downstream to show you how we envision this technology being potentially very useful in practice. So the first application I want to show is a container closure study. We're taking a fixed monoclonal, we're using a fixed polyclonal antibody in this particular case, and we're stressing it in 2 different glass vials. One we're going to call a generic glass vial, which is very cheap. The other one's going to be a plasma-coated shot glass vial, which is engineered to be a little more friendly to biologics. And we're basically going to stress the proteins solutions in these two different vials the exact same way. The only difference is the container closure interaction, and see if we can see differences in the particles that come out of this particular study. And so the question is, how many images do we need? Can we tell the difference between the two if we do this? And how many images do we need to statistically distinguish between the two different types of container closures? So shown here are the fingerprints that we get from generic glass. So you get this kind of fingerprint that shows up here. And our reference condition is going to be the plasma-coated glass. So this is the fingerprint that you get from that. Once you've estimated your fingerprint, what you can do is you can take a collection of images and do this image compression scheme. You can also, if you have a large collection of images like you do in formulation when you're doing accelerated stress tests, you don't just have to do the point evaluations. You can actually get a fingerprint for your new case. So here's the fingerprint that you have for the generic glass study. And if you look at these two, the shapes are very similar, but the mode's different. So this one in the generic glass, the particles are really concentrated towards this area, or the compressed embeddings of the particle images are concentrated in this area, whereas it's a little more diffuse, or it's spread out more in the plasma-coated glass case. If you look at different regions, though, the particles that correspond to different regions, we have found that oftentimes the morphology, the human interpretable morphology, are very similar. And so if you look at the mode of this PDF, in these two cases, the particle types that show up near the mode are very similar. And so what happens with this particular glass case, it seems like you just form more of these types of fuzzy white particles in the generic glass, whereas you form less of them in the higher quality plasma-coated glass. So it's a population shift. This is all well and good. That's a subtle change, so you can detect that subtle change, but you can also look for large changes. So I want to stress, again, how we really envision using this tool in kind of a broader context. So imagine you have your PDF, your reference PDF that you've established your baseline, and so that's this fixed PDF that you look at here. You then have a small number of images that you gather from your PAT line or from your formulation study, and you have these images and you compress them down to their two-dimensional representation. So here we have a small number of images here, less than 100. We have about 5 times 20, right? So each of these images gets mapped to a point. And so the question we have is, how consistent is this distribution of points with the baseline PDF or the fingerprint? And that's why we estimate the PDF in this particular case, because it allows us to do formal hypothesis testing. So from this collection of points, which I'll show later the mechanics of this, but we can formulate a metric for quantifying the similarity of these particles or the consistency of this distribution of scatter plot points to the PDF, and that results in a test statistic. And so for this particular case, for this 20 set of points, this is what the test statistic value was. Since we're doing test statistics, if we repeated this procedure over and over again, what we would do is we get a test statistic distribution under the null or under the baseline condition, and this is the distribution we expect when everything's going well. And so anything above this line is, we have a 95% chance of detecting something going wrong, or 5% of the time it just happens because of chance. This is the standard type 1 error type threshold, right? So in the case where you're sampling from the actual baseline, you get a test statistic value that's small, less than the threshold value. In the case where you have a new process upset that you've never seen before, you can still evaluate the images, do the same kind of scatter plot evaluation. They start to pile up in areas that are not probable, consistent with your baseline distribution. In that case, your test statistic, any reasonable test statistic, should result in a large value, shown here, which is well above the threshold for hypothesis testing, and you reject the hypothesis that these points came from this distribution. So that's the idea here. So the idea is you summarize this small collection of images with a single metric, which is a test statistic. And there's a variety of ways of doing that. But the idea is that you can repeat the procedure over and over again. So in the glass container closure study, what we were looking at was about 200 images at a time in that particular study. When we sampled images 200 at a time, we'd evaluate the test statistic. We'd repeat that over and over again. And this is a histogram of test statistics that we get when we sample from the baseline condition or the fingerprint or the null. And this is very consistent with basically a normal PDF, very close to it. If you get this case where the mode shift, remember in the generic glass, the mode was peaked, the test statistic distribution starts to shift very subtly. So you start to reject about 30%, even with only 200 images. You know that there's not consistency between the fingerprint of the baseline, the expensive plasma-coated glass, and the regular glass. So with only 200 images, you can start to see differences between the aggregates that were formed, where the only difference between the protocol was the container closure interaction. So you can quantify container compatibility. And here is kind of a more obvious application of this technology where you have a reference condition that's coming from a pump, and you have a new pump that is, say, you have a pump in New Jersey, and you have a pump in Puerto Rico, and you'd like to see if the particles that are produced from these two pumps are consistent with the fingerprint. And here's the fingerprint is the exact same in both images. What we're showing are the compression scheme, the embeddings for the two images shown above. If you look at these images, a human has a very hard time distinguishing between these two images. But in this fingerprinting approach, you start to very clearly see that something is awry. These things are clearly not sampling from the same PDF. And with our goodness of fit hypothesis testing, we can quantify that with various sample sizes. So shown here is just kind of summarizing how well we can do with this type of approach when, if you give images from known training conditions or the baseline or whatever you've picked there, you don't reject more than you expect. You achieve your type 1 error rate. You have n equals 20, n equals 100. Everything's checking out fine. So you're rejecting, if your threshold is, I want to make an error 5% of the time, what we're showing here is the fraction of cases that are rejected in the particular hypothesis testing analysis. What you're doing is we're still freezing this reference null condition where we're looking from a pump, but if we put in conditions like a mechanical agitation or a viral clearance type particle, we can very easily see with 20 particles that there is just not a consistency between these two distributions. And this is a way of quantifying that formally. With 100 particles, you can get 100% rejection. You can detect it perfectly. What I want to stress, though, is you can detect conditions that you did not train the network for. So you can have pump shocks from a new plant that produce new types of particles, but you didn't include that in your network. So this technique is really an outlier detection tool. It's very powerful for detecting upsets that you did not account for early on. And so that's what I want to stress. These conditions were used in the training of the compression scheme, these three conditions, whereas this was not. So this was a completely novel class. This is like the snake class in the example we showed before. But we're still able to detect it with 100% accuracy, basically with 100% accuracy with n equals 20 or n equals 100 images of these fuzzy flow imaging microscopy images. So it's a really powerful tool for quality control. In terms of time, we're going, since we waited a little bit, I'm going to go through this really quickly, but anybody's interested in the details, I'd be more than happy to talk about. But the idea is you have a complex distribution of points in your PDF. So if you have your baseline condition, those embeddings are spatially correlated. They're not independent dimensions in the raw dimensions. The advantage of estimating the PDF, going through the trouble of that, is once you have a PDF, you can then introduce new transformations that make hypothesis testing easier. And that's the bottom line. You take a correlated collection of nonlinear, complicated distributions, you standardize it or normalize it to a collection of new random variables that have a known distribution that's simpler than in this particular case. What we're using here is the Rosenblatt transform. So it takes a collection of images, or 2D points, and transforms them to the unit square under the correctly specified model. And so what you're really doing is you're taking this collection of points, you're applying a nonlinear transformation to these points, and if the thing is correctly specified, it should fill out this unit square. And as you get more and more samples, it should just be a nice uniform flat PDF. So you kind of standardize your analysis. When points come from a different distribution, you start to go very far away from that kind of uniform IID normal distribution on the two dimensions. So it helps you detect outliers independent of what the underlying shape of your baseline is. So it's kind of a normalization technique. You could think of it that way. But then there's a lot of other tools that you can leverage in that particular case. For checking, are these points actually consistent with a uniform distribution? This is one example of a technique you can use for quantifying the similarity of these scatterplot points to a uniform distribution. And the question you're asking is, if I had an infinite number of particles, this is what I should see. Are these fluctuations just statistical noise? And that's what this test statistic quantifies. So you basically are integrating the differences, the deviations between the large sample limit and what you observe empirically, and you're computing a metric, and that metric is your goodness of fit test statistic. When you have a sample that is clearly not from the uniform distribution on both sides, these large parts under the area, integrate, and result in a large test statistic. And that kind of gets us to that's how we quantify that in the previous case when we're looking at the glass container closure study, just for those of you who are interested. But I want to change gears a little towards validation. So another issue in this technique is these instruments are very sensitive to a variety of optical parameters and conditions, preparation conditions. We need to start validating this technique if we want to use it for more quantitative studies. And this is a study in collaboration with the FDA and NIST folks. What we're envisioning considering using is a polymer surrogate standard. So ETFE is a polymer that was engineered to have a lot of optical characteristics similar to MAB, protein aggregates. And so this is being used to help in addressing a lot of issues that come up in calibrating flow imaging microscopy images. But we want to use it in a slightly different context. It's been historically used a lot for sizing and counting. We want to use it as a tool for assessing how consistent are your measurements. And so that's what we're going to be, and it has other use cases. You can see that the images here, these are the ETFE images. They kind of look like protein particles, but the algorithm has an easy time separating these two when you use the full information content in the images. What we're really doing is we're trying to characterize fingerprints of these various, either the protein or the ETFE particles. And so what we want to see is how consistent between vials are these fingerprints that we're estimating. So what I'm going to show first is looking at a gamma globulin study from the FDA folks. They mechanically agitated this particular protein, and the reference vial we have is shown in green. Two things to note in this particular plot are there's two modes of this distribution. One is what's shown in this particular plot is if you look at these contours, remember this is a PDF. So what the 10% here shows that 10% of the probability mass is contained in this area. If you go to the 30% contour, 30% of the probability mass is contained in this area and this area. And then as you go out to the larger numbers, 80 I believe here, if I can see correctly, 80% of the probability mass is shown here. But there's two modes, there's two peaks in this distribution. And these two peaks, interestingly, correspond to two different types of particle morphologies. One, the darker types of particles shown here, and here are the lighter type particles shown on the top on the right. The darker ones are, of course, on the bottom right. I don't believe my pointer is actually showing up on your end. But that's interesting that the morphology maps to two different regions. But what we're really interested in this study is when we take a new vial and estimate the PDF from a large collection of new vial data, there's basically perfect overlap between the mechanically agitated gamma globulin from the reference condition and from the new vial. We repeat that same procedure again with another test vial, and we get not exactly perfect. There's some subtle differences, but we're getting a very consistent picture of what particles look like in this particular container closure for this particular stress protocol. This is great. This lends us to believe that this can be used possibly as a tool for lot release or quality control, or even other studies that we'll talk about later. But what I also want to show is proteins are expensive, they're hard to sample, and they're finicky over time. It's hard to get a good protocol sometimes to actually get very consistent particles when you stress them. ETFE is a polymer, and it's not reactive with itself, and it's very shelf stable. And so that's why we want to use it as a tool for, if you reject something, you probably want to go back to your microscope to make sure that it's consistent with its past measurements. So suppose you go through and you see a lot release and all of a sudden the test statistic value becomes very large. Instead of just shutting down the plant, one thing you might want to do is use a reference material that has another fingerprint. For example, ETFE has a fingerprint where we measured it in 2017. And then we take a sample from 3 years later, and lo and behold, the samples have very similar fingerprints if the microscope's correctly calibrated. So if you reject the protein and you see that the ETFE lines up, you can be very confident that something's wrong with the protein or the production process. However, if you go through this exercise and you notice that the ETFE doesn't line up, then you know there might be something wrong with your optics, which happens with this particular measurement modality. So it's nice to know why you're rejecting these particular cases. And so one study that we're doing in a forthcoming publication, or a publication that we're going to be submitting, or a manuscript that we're submitting, is how sensitive is this approach to some tunable optics. And so what we did here is we took a collection of NIST ETFE and we measured a fingerprint. So that fingerprint shows up over and over again as the dashed blue lines in each of these particular panels. So this is a fixed fingerprint from one day from a large run where we measured about 100,000 particles of ETFE through the flow cam with a particular set of optic settings. There's a lot of tunable parameters in that. It's fixed though. Then what we did is on a separate day from the same lot of ETFE, the only thing that it was a new day, so we fired up the folks at NIST fired up the microscope and measured things on a new day from with a new injection in the flow cell, but they were systematically changed the stage position. So the stage position for the flow cell basically detects if it's in focus or not. And what we did is we then collected a large number of particles from those studies, and we were mapping the fingerprint that results in black from those studies. And what you can see is the fingerprint's not that far off, but you would reject this easily, 165, 155, and we'll quantify that in the next slide. But eventually you start to get very close overlap. So again, this is consistent, showing that your optic settings were the same. In this particular case, stage position 145 was the most consistent with the previous data that we looked at. And so, again, we can use goodness of fit hypothesis testing to not just qualitatively look at that, but we can quantify that, the minimum of these rejection fractions. So if you were to take these samples 200 at a time and say how many do you reject when you're at stage position 45, it's very close to what the nominal type 1 error rate is. So that's wonderful. So we've got tools that can go towards validation. But I also want to go through really quickly another protein study for shipping. A lot of things can go wrong when you ship a protein product. It goes through a truck, it might go through air. Some of these jovial fellows might toss your product, even though it says to handle with care. Lots of abuses can occur. So the question is often, what are the most important, what kind of forces cause aggregates in this kind of case? And so again, I'm going to go through this really quickly because of the time. But what we're going to be doing is it was noticed in a particular study that was carried out by the 10 Radnoff lab, where they were actually interested in looking at different packaging. So what they did was for this particular study was they had two types of packaging, one that they call the shock absorbing packaging and the other they call the lab packaging. So the shock absorbing packaging was a fancy packaging that was supposed to minimize impact of really, of abuses in shipping. The lab packaging was something that graduates didn't do. Imagine just duct taping a bunch of vials together in a package and sending it in USPS. But we didn't just do that. We prepared two different packages. Ted's lab prepared two different packages of the exact same IVIG material, but it was sent to North Carolina by FedEx and then came back to Colorado on UPS. But there was kind of a covert operation. What they did is in the packages, they also put force monitors to monitor the G-forces that these things experienced. And we actually skipped the G-forces that you see, but you see large spikes, which are shocks. We basically think of it as being dropped, but there's also low frequency amplitude variations that you see that kind of correspond to mechanical vibrations that you might experience in a truck. There's also a variety of headspaces used in this particular case. In this particular, what we're showing here are the number of particles that were produced as the headspace increases, or the amount. So in the left shown here is the lab packaging and the SAP packaging. This is all filled. When you fill the thing, there's very few particles that form, but as you give more headspace, more things can slosh. The packaging had no influence on the particle count, which is interesting. As you get more headspace, the thing just sloshed around and you get more particles independent of the packaging type that you look at. So that inspired Ted's lab to do a study where they did a simulated kind of accelerated stability study where they used a plate shaker to simulate the vibrations that you observe on a truck, and they basically put the thing in a dryer or a tumbler to mimic the large impact G-forces that we're looking at. And so the idea is now we can, before we were sending these packages out into the wild, we have no idea what happened to them. We just had force monitors that measured what happened. Now we have a very controlled study where this is the force protocol that this particular package experienced, and this is kind of low amplitude vibrations that they experienced, and we know what we did to them. That's interesting because we can look at the the packaging that we looked at before, where the counts were changed, we can apply this fingerprinting approach. And the take home here is the fingerprint doesn't change as you go over time. This is the wild data from the shipping data. The fingerprint is independent of the packaging, and what this suggests is that the mechanism of formation does not change. You're getting the exact same types of particles that you're looking at, but the counts are just increasing. So you're producing more of them. That's what this particular study suggests. What the advantage of this particular study was the shaking that we did in the lab and the tumbling, which we know what these stress protocols look like. We can apply these fingerprints. The fingerprints for the shaking look like this. We know the stress history that we applied here. The shipping, which we have no idea what happened to the shipping particles beyond the force measurements, the shipping looks a lot like the fingerprint of the shaking, which suggests that the low amplitude shaking of the truck is the predominant cause of the aggregates that we see. So that type of that high frequency shaking, but not high amplitude force shocks. This is kind of a root cause analysis. So we're not looking at quantitative goodness of fit. We're basically just doing a qualitative study where we did known stresses to the particular product, the IVIG, with tumbling and shaking, estimated the fingerprint, and then applied the fingerprint to a wild study, the shipping study, and we see that the particles that were formed, the embeddings, the distribution look very much like the shaking. So again, it's a useful exploratory tool. And I have presented a new method for characterize using CNNs and tailored it to issues that you see in protein therapeutics. The method exploits morphology that's encoded in the images. We're making better use. We're going beyond count and size. It can handle unanticipated stress conditions. You do not need to prepare for every known stress condition that might happen. For example, in the shipping study or the pump experiment that we looked at before where the plant was not included in the model, which is very useful in practice. The validation studies we show with NIST are underway. We're still exploring that. We're still researching that as well. And we're also applying this to other imaging modalities. With that, I'd like to pass it over to Sandy to talk a little about the product that's in development.
Thanks, Chris. Now that we have a detailed understanding of the technology behind particle sentry AI, let's talk a bit about where we're at with respect to releasing this technology to the marketplace. So Chris showed this slide a bit ago. While we're very early in the release process for particle sentry AI, you can see it has wide application across the entire drug product life cycle, from early discovery and development to post-release and surveillance. Particle sentry AI can help you detect subvisible particles within your drug product, identify the causes for those particles, and provide actionable data to control their propagation. In brief, our capabilities include fingerprint training and analysis, much like what Chris has shown, batch comparisons, stress studies, and any of these activities are an excellent place for us to begin to work with you to learn how particle sentry AI can help characterize your proteinaceous drug formulation or products. Because our technology uses images from commonly used orthogonal techniques like flow imaging, microscopy, we can also do retrospective analyses of any collection of existing images. We can also work to develop fingerprints that include many common stress conditions and help put you in an excellent position to begin using particle sentry AI software once it's released in Q4 of this year. In summary, today we process images on your behalf, and our fingerprint development and training software is completed for this purpose. We're gathering feedback to finalize the user interface for particle sentry AI to be released in Q4 of 2021. Testing to support validation activities in compliance with USP 1225 validation of compendial procedures will be completed in Q3 of this year. And one paper based on our technology has already been released this year. You can see that on our website, which is www.sentrysci, sentrysci.com. And that one's case study 4 on shipping and vibration-related aggregation. And we anticipate at least 2 more publications demonstrating the utility of particle-centric AI later on this year. We're ready to engage with customers to see how we can help you detect subvisible particles within your drug product, identify the causes for those particles, and provide actionable data to control them. With that, I'd like to thank, on behalf of everyone here at Century Sciences, I'd like to thank you for participating today and listening to Chris's detailed explanation about this technology. We're hoping to answer some good questions. So with that, I'll turn things over to Elizabeth so that we can get to your questions.
Thank you very much, Sandy and Chris. We have quite a few questions. Our first one is from Taylor. Is it possible to change a fingerprint once it's created?
Yes, it is. There's a variety of ways you can do that. You can retrain the network completely from scratch if you have a new set of conditions that you'd like to incorporate. Or there's other techniques where you can kind of freeze the conditions you had before and continuously morph or add new information in to try to keep the old fingerprints in the same position. So those are 2 options. It just requires having a new set of training images that are labeled that you know where they came from. So yeah, it's rather easy.
All right. Glenn asks, at our company, we have people that review images and look for outliers. What's wrong with that approach?
Yeah, one of the themes of this talk was I tried to use the dog and cat analogy and then showing how complex the morphology is in these images. Even subject matter experts have a really hard time finding good quantitative and automatic features that are embedded in the images to make decisions on. So this technique is a really systematic, consistent way of assessing the particles that are coming out of these images. Even though they might be hard for a human to interpret, this is a algorithm that is going through and processing in the exact same way. It's not like someone gets tired in a visual inspection type thing where humans are looking at this. This is a completely digital automated technique, and we see that being an advantage of going over humans looking for outliers.
All right. We have 2 questions from Dib. The first question, which file format is used as input? Is it .png?
That doesn't really matter too much. It's a matter of, we like to try to use lossless formats. I mean, the FlowCam uses TIFFs. That's where we extract from. But you just need to be consistent with all the images. You need to have the same resolution, whatever you're doing, whenever you're comparing these types of tools that we're looking at. You just need to make sure that you have the same exact image type and compression scheme that you used consistent throughout. It doesn't really matter what the format is. It's just a pre-processing step to pull it out. But you can't start mixing and matching. That would be a bad idea in general, because eventually you'd like to do testing. And if you test on a PNG that had one type of format, then you go to a JPEG, for example, that has that loses some information, that's not a good idea. So.
All right. And the second question, how are you extracting the particle images?
So could that question be clarified? Is this from from a panelist? So extracting, so the device, the imaging device, the flow imaging device is measuring these. So they're basically, you've got a vial of product that you'd like to inject through the FlowCam in our particular case. You have a syringe and then you inject that into the flow cell. Then it just measures all the particles that are in the stream in the flow cell, images each one of them, and then that's how we get our input data for these techniques.
Okay. Our next question. Sorry, I'm sorting down through the questions here, is from SP. Can the software be used with MFI images? What's your experience in terms of correlation between similar particles across different instruments?
Yeah, that's a great question. So the measurement modality, these things are very sensitive to subtle features in the imaging modality. As we showed with the ETFE, just changing the focus can change the fingerprint. And so each of these MFI, yes, this is applicable to MFI, but you can't really compare MFI images to FlowChem images on the same product. They will definitely differ. So when you're doing this technique, you need to lock down your instrument that you're interested in looking at. And each of them have different advantages in terms of their camera resolution and other tunable parameters. But in theory, yes, this could all be applied to MFI, and we have used it in the past, applied it to MFI. We just haven't published it.
All right, our next question is also from Dib. What is needed to obtain a fingerprint?
So the only thing you need are a large collection of labeled images. You need your baseline condition, but it also helps to use other images from stress conditions. So think of having an accelerated stability study where you have a product that might have come from a particular stress condition, but you do light shocks, you do mechanical agitation, you do freeze thaw. Those all might be included in your fingerprint construction, because the more you can show the types of stresses that the thing might occur, the more power this embedding technique has in representing different possibilities. And so what we're really interested in is trying to get fill finish data for a lot of these things, a historical collection of fill finish data that should be from a clean room and very, very, very reproducible. But then you could also compare that to known stress protocols, and you can include all of that in your fingerprint construction. So we're trying to leverage all the data that needs to be done for a drug product application anyway, and trying to use that in the fingerprint construction.
Thank you. Our next question is from DP. What kind of output does your software provide in addition to the fingerprint?
Yeah, so in addition to the fingerprint, the current version of the software outputs test statistic values. Corresponding p-values are for those test statistic values that are a little more intuitive. We can also graphically visualize things in a variety of ways beyond just showing the PDF or the fingerprint shown as a contour plot. We can plot the embedding points on that reference PDF, but we can also estimate a new probability density function for the embeddings that come from your new condition. There's a lot of visualization tools that can kind of help in quality control charts qualitatively, but also quantitatively. So there's a variety of outputs that are in the initial version of this.
All right, thank you so much. That appears to be the last of the questions, so I would like to take this opportunity to thank our presenters, Sandy Hutchison and Chris Calderon. I'd also like to thank the folks at Century Sciences LLC for sponsoring today's event. On behalf of Cambridge Health Tech Institute's Global Web Symposia series, I'd like to thank you all so very much for spending this time with us today, and we hope to see you at future webinars. Thank you all so much, and have a great day.